My name is Muhammad Farhan bin Nasir, metric number E22A0158. I will be presenting the introduction for this study. First of all, let me get on the introduction or background of this study by first knowing what is insect. Insect is the largest and the most diverse group of organisms belong to the class Insecta, with their segmented body, exoskeleton, and remarkable adaptability. They play a crucial ecological role such as pollinator, nutrient cycling, and serving as a prey for higher trophic level. And from the family Formicid stand out for their social organization and ecological dominance, also the extensive distribution. The prime example is the Asian river ant, Oecophylla smaragdina, a prominent species in tropical and subtropical ecosystem, known for their complex social structure and cooperative behavior. These ants play a vital role in maintaining ecological balance. And our research focuses on studying the population the Asian river ant in forested and agricultural habitat at Jeli, Kelantan. This research aims to bridge the gap between the research and conservation practice, highlighting the ecological importance of Asian river ant. Our target audience include the student, researcher, and agricultural practitioner, emphasizing the conservation value of species and its broader ecological significance. By understanding the Asian river ant, we can inspire better conservation strategy and promote sustainable ecosystem management. For the problem statement, the problem statement for this research is the limited information on its population dynamic and habitat preference in specific localities like Jeli. The difference between forested and agricultural habitat may affect the colony size distribution and reproductive success. The objective for this research is the first one, to compare the population density and colony characteristics of Asian river ant in forested and agricultural habitat at Jeli. And the second one is to determine the environmental factors that influence the establishment and success of Asian river ant colony.
The Asian river ant is an ecological significant insect that thrives in diverse habitats, including forests and agricultural landscape. Its unique behavior, such as nest weaving and cooperative colony management. Firstly, ecological role. The Asian river ant was a prominent tree-dwelling species famous for its elaborate left nest and crucial ecological function in both forest and agricultural environment. According to Marcella Pimi 2019, these are common in treetops where they display aggressive hunting behavior that aid in natural pest management, improving plant health and fostering biodiversity. Their feeding behavior is affected by environmental conditions like temperature and humidity, according to Kumaran Teban 2019. This attraction underscores the species' role as a keystone in maintaining ecological balance. The second one is population dynamic. River ants are interactive tied to their social structure and polydominous colony organization, where multiplying nests and interconnect by extensive trail system. Colony size and nest distribution depend on habitat type, resource availability, and interspecies competition by, according to Peng and Christian 2005. In forested habitat, the structural complexity provide stable, rescue nest site, fostering large and stable colonies. The third one is habitat preference. River ants exhibit distinct habitat preference that significantly influence their distribution, colony size, and behavior. Forested ecosystem, with their high structural complexity, diverse microclimate, and abundant resources, provide optimum nesting conditions, fostering large and more stable colonies, also promoting long-term colony growth and persistence. This environment also supports a diverse prey base, enhancing the ant ecological success. The fourth one is foraging behavior. Foraging behaviors of river ants showcase remarkable flexibility and effectiveness in gathering resources, marked by the creation of both foraging paths and high collaborative tactics of optimized resources use. These ants depend on their interacted communication method that use pheromones and touch signal to organize group tasks and effectively find, move, and store food supply. Elements like prey abundance, habitat configuration, environmental factors, and and interspecies competition are vital in influencing their foraging behavior. Last one is interaction with human agriculture. Weber and have maintained a long-standing symbiotic association with human farming method, especially in South Asia, where they have a sufficient biological control agent for 100 of years. According to Van Mel 2008, they targeted various pest species, aiding in the health and productivity of fruit approach, particularly in mango and cashew palms.
This research will be conducted at Jeli, focusing on two distinct habitats, which is a natural forested area near campus and then agriculture areas such as crop field. The materials that we are going to use in this research is binocular, field notebook, GPS device, measuring tape, digital thermometer, hygrometer, transparent collection jar, and digital scale. In each habitat, we established 5 transects, each 100 meters long and spaced 50 meters apart to avoid overlap. For observing ant colonies, we use essential equipment including binoculars, a GPS device, a measuring tape, and tools to record the temperature and humidity. We will also record the microclimatic condition within 2 meters of each colony, including temperature and humidity using a digital thermometer and hygrometer, and habitat characteristics like vegetation type and canopy cover. To understand foraging behavior, we observe each colony for 10 minutes, focusing on the direction of foraging trails, the type of resource collected such as prey or non-prey items, and number of active workers involved. And then we will capture prey items, store them in collection jar, and later weigh them to analyze the average size and biomass. In data analysis, our first step was recording key variables in a spreadsheet. This included habitat type, which is forested or agricultural. The second one is colony size and density. The third one is microclimatic conditions, such as temperature and humidity. And the last one is foraging behavior, that focusing on prey type and prey biomass. By systematically collecting this data, we could analyze the relationship between these factors such as habitat type. To compare the colony density and size, we started by calculating colony density using the formula provided in our methodology. We then applied a t-test to compare the average size and the density of colonies in forested versus agricultural habitats. This allowed us to determine if there were statistically significant differences between the two environments. Next, we analyze microclimatic condition. Using Pearson correlation, we assess the relationship between colony density and environmental factors, specifically temperature and humidity. This helps us to understand how changes in microclimate might influence ant colonies in different habitats. While for foraging behavior, we wanted to see if the type of resources used varied between habitats. We performed a chi-square test to compare the utilization of prey versus honeydew in forested and agricultural areas. This analysis gave us insights into how habitat type impacts resource selection. Finally, we examined prey biomass. A Mann-Whitney U test was used to compare prey biomass between the two habitats. This non-parametric test was ideal for detecting differences in prey availability and utilization across these distinct environments.
Hi, my name is Lily Anaribau. My metric number is E22A0192. Here, I want to present about research outcome for population biology of Asian weaver ant with scientific name Oecophylla smaragdina in forested and agricultural habitat at Jeli. Here I have a few research outcomes that we found. First, colony density and habitat preference. Colony Oecophylla smaragdina in agricultural habitat are anticipated show the higher density compared to forested habitat because potentially due to availability of resources like plant, excretion, and prey diversity. And forested habitats colonies will demonstrate greater spatial dispersion and dependency on natural vegetation for nest building. Second, influence of microclimatic factor on colony dynamics. Positive correlation expected between colony density and meteorite temperature. 25 to 30 target Celsius and relative humidity level 16 to 80% in both habitats. Forested habitat have greater climatic stability, fostering large colony compared agriculture area. Furthermore, foraging behavior and resources utilization agriculture habitat. Foraging provides direct or indirect nutritional benefits such as pest, insect, and extraflora nesters. Prey biomass analysis higher reliance on some solar-based species in agriculture area, contributing to pest control benefit. Forested habitats show a balanced utilization and plants drive resources due to diverse vegetation structure. And last but not least, application for ecosystem service. Oecophylla smaragdina biological control agent in agriculture setting, offering insight into sustainable pest management practice. Habitat management strategies could promote Oecophylla smaragdina population for ecological and agricultural benefit. These outcomes are expected to bridge knowledge gaps regarding the adaptability and ecological role of Oecophylla smaragdina across habitat, providing a foundation to further research and practical application in conservation biology.